Okay, everyone, it's March 31st, so we've got about 5 more weeks of a course. So we're putting everything into Blackboard. I've had a few students who have not done a few of the assignments, specifically in lecture, the test, and in lab. The lab assignments, so I don't know what to do at this point, so I guess I'm going to post everything and let you guys do them whenever you can. I know things are coming up, but when I get a 3 or 4 week delay with no activity, that doesn't send a really good flag to me. So trying to be as fair as possible. So you guys knew the course at your pace. I know it's not optimal, but moving forward, you're gonna have to snap out of this, or shouldn't say snap out of it, but you're gonna have to figure out how we're gonna move forward. Hopefully, this will end by the fall. I'm not really sure, but I have put a lot of the questions in lab and again in lecture. So in case you haven't figured it out yet, if you do the lab well, very similar questions will be on the lecture, so you all should be able to do well. And all of these questions are very, very similar to what you're going to see or be tested on in the future. So spend a little time in mastering, go through the videos they have there. I know you may think that 7 or 10 minutes is arduous or whatever, but just I want to remind everyone that when my generation went to school, we didn't have Google and we didn't have Wikipedia, so we literally had to go and carry books around, go to the library, and spend hours. I mean, in grad school, I can't even tell you the amount of hours I spent, and I don't want to give you the sob story, but the internet was relatively new in 1999. It was there, but nowhere near what you guys have now. So that's the end of my my dissertation on that. So let's spend some time on antimicrobial therapy. And remember too that a lot of this is going to be very similar to antimicrobial as far as detergents and ways we disinfect surfaces, or skin, or if we want to sterilize something for intercavity use in the body. And we'll be going over some of the interactions of how these work. I'm not going to have you guys memorize very, very specific antimicrobials because they're changing and things are becoming resistant to it. However, you should know, and they will be testing you on classes of drugs. So certain classes of drugs, how do they work? What is their MOA? What is their mode of action, which is probably the most important. Okay, and then you have to realize you're going to be, you may not be prescribing unless you're an MP or PA or whatever. You may be administering these antimicrobials, and you have to figure out, well, this microbe might be more susceptible to this antimicrobial. However, the patient is old, feeble, dehydrated, they have an immune compromised system. The patient is gravid, all right, pregnant. Or maybe they have a reaction to they've had a reaction to the antimicrobial in the past. And please remember too, before I forget, when you go to pick up a prescription and that pharmacist, or God forbid you should get a pharmacist, the assistant, whatever, to your course they have or whatever, not to diminish it, but is asking you, have you ever had a reaction to medication before? And you're gonna obviously gonna say no. If you did, you'd tell your doctor. But please realize, if you have a drug reaction to a medication, it's never the first time. Right? It's always the second or third time, because remember, you don't have your B cells are naive to it, and you don't have the antibodies to it. You don't have the activated CD8 T cells for that. So just kind of remember, you give someone a medication, it may look might work great the first time. Be very, very effective, but the second time you take it, you have an allergic reaction, and people are like, well, why am I having a reaction to this? It can't be the medication. I've taken it before. So just kind of keep that in the back of your mind. But we want to administer a drug to an infected person that destroys the infective agent without harming the host cell. So we're going to actually figure out what makes bacteria different than us or prokaryotes from eukaryotes. So I know I went over that before, but we're going to hone in on those specific differences. We're going to hijack it, or we're going to use it against the cell. So that is called selective toxicity. We definitely want to wipe out the agent. Sorry, guys. Bodie, no. Sorry, dog's going crazy. Antimicrobial drugs are not produced naturally. I'm sorry, they're produced naturally and synthetically. So we're going to see that Streptomyces and some of these Clostridiums are going to be giving off substances that inhibit growth around their Bodie, no, around their environment to inhibit drug or other microbes growing around them, right? Right, and we're going to do them. We're going to grow them synthetically. Number one, it's faster and less expensive. And with a lot of these other drugs, we have to keep changing that side chain because bacteria are very smart. They're going to figure out a way of getting around, whether it's a efflux pump or penicillinase, or they'll change their binding site, or they'll change their metabolic pathway, like in sulfa drugs. And we have to keep refiguring out new ways to get the antimicrobial into the bacterium, because they'll find a way of resisting it. So selectively toxic to microbes, but non-toxic to host cells, right? So I put something similar to that in your lab, in your test, 3 or 4 questions, very, very similar. So, chances are you're going to get a question on that. And remember, everything's randomly generated, but there's 4 or 5 ways they're going to ask the same question. So you'll just get one one version of that. All right. Microcidal rather than microbiostatic. So is the antimicrobial going to actually kill the bacteria? It doesn't have to kill it with hand-to-hand combat or kill it immediately, as long as it can make sure it can't produce proteins or ribosomes, or if it can cause it to produce defective proteins, any defective protein, eventually it's going to die, all right? Because the microbes are going to be making proteins or making enzymes that it needs for viability. So if we can alter the bonding site, or give it an analog or something that would, put jam up the gears or whatever, shall we say, it'll work. And or is it microbiostatic? Is it going to really slow down its replication so that your immune system has a way of fighting or interacting with it? It has to be relatively soluble, all right? Functions even when highly diluted in body fluid. So remember, we have to be able to take these orally or intramuscularly in the deltoid or wherever we're taking it. But if it is not soluble, we can't get it into blood plasma, we can't get it into the interstitial fluid. And without getting into the interstitial fluid, we can't get it to the causative agent, right? And some of these antimicrobials like nystatin and some of these fungal medications are going to be very, very toxic to human cells, because they're going to be working on the biphospholipid layer. Now they're, we have cholesterol and sterols, and they have estradiol in their lipid layer or their cell membrane, but it's very, very similar. So some of these, they might be soluble in water, or they might be soluble in fat, or maybe they're amphoteric, which means they're soluble in either, right? They go either way, per se. All right, remains potent long enough to act and is not broken down or excreted maturely. So it has to be viable or active long enough to get to the system, or get it to the area of infection or area of cause. But we also don't want it to get broken down very, very quickly. And we have to make sure that it can be broken down by the liver and the kidneys, right? And that's why we dose things out. We dose it out 4, 6, or 8 hours, or 2 hours, or whatever, because we have to figure out, well, how long would it take that substance to be broken down? We wouldn't want something antimicrobial in our system for days, weeks, or a decade. If it's not being able to broke down, that's probably not a great idea. Right? It doesn't lead to the development of antimicrobial resistance, so we don't want the antimicrobial to make enzymes that could break it down or block the, that could block the binding sites. It could change the shape of the binding sites. It could have more efflux pumps to pump it back out. It may be able to change its metabolic pathway. So we'll be coming up on some of those things that they can do. And remember, these microbes, I said, if there was a clock of the world from midnight to midnight, we literally showed up at 11:59 p.m. I mean, these bacteria have been around for way longer than we have, and they're going to figure out, no matter what we do, no matter how smart we are, they will find a way around it, either through gene expression, through plasmids, from the fertility plasmids, or through just random mutations. And please also realize, without getting super controversial, that a lot of these viruses out there are literally these bacteriophages, and these viruses, as lethal as they may be, or as sick as they do make us, there's a lot of talk and study on how they're literally upgrading or uploading our genetics. And people will say, well, we have a bunch of junk DNA. Well, it's not junk DNA, it just hasn't been expressed. And I mentioned before in one of the videos, I think your environment and your thoughts is gonna cause your gene expression. So if you're at all interested in that, spend 15 minutes and listen to anything by Bruce Lipton. He'll actually tell you. You can put a stem cell in any environment, depending on what you do to it, as far as music or energy, music, thoughts, or whatever. It can change what that cell will produce. And if you want to see something called the secret of water, water is our plasma. Our body's made of 70% water, and water is the only I won't get too deep here, quantum physics. Water is the only substance that can actually hold information and hold energy. So if you watch The Secrets of Water, they can take water, you can expose it to whatever, love, hate, jealousy, whatever. And then you literally take that water and you freeze it. And then you look at the crystalline structure. And every single time you look at that crystalline structure, depending on what that water was exposed to, it's different shape. Now, I mean, I know every snowflake or every ice crystal is different, but if you look at these images, take 3 minutes when you're taking notes, go in and see The Secrets of Water. Asimoto, I think his name is, or I think he's Japanese. But look and just see like how water exposed to joy or love looks, as opposed to something that is exposed to hatred or rage or wrath or something like that. Now I'm digress. No wonder these videos take 2 hours. Anyway, complements or assists the activities of a host defenses. So my thought process is I would rather not take any medication, and I really would rather not be vaccinated and guess it's something that I think is detrimental. Polio, measles, mumps, rubella, diphtheria, rabies. If there was an episode, not an episode, if there was an epidemic of bubonic plague or tumaria from rabbit urine or something, weird that I'm thinking of, there was a vaccine, I'd be taking it. As of right now, I'll keep my thoughts to myself as far as what's going on right now, but I am going to prefer, personally, to be exposed to whatever is in the environment, and for my body to make whatever antibodies it needs through my B cells. Now, whether they've been activated before from a prior SARS virus, or if it's something new or novel, or, everyone's talking about this new mutant UK strain or whatever. So, now we can't call it the China virus. Now, we can call it the UK virus or the Australia virus or whatever we want. And then, we'll be defined as, all right, we won't get into that. Okay. So we want it to remain active in tissues and body fluids. We don't want it to be inactivated, right? Like, we talked about the hypochlorites and some of these disinfectants were inactivated by organic material. Well, we don't want these medications or these, I'm going to call them chemotherapeutic agents. So when you hear that word, you may be thinking of like cancer drugs, but these are all chemotherapeutic. So they're chemicals that have a therapeutic effect, right? Reason are readily available to the site of infection. So we want to be able to get it to the inside. So whether we're injecting it or if it's a really bad case of sitting for fringes and we have to go in and debride that dead necrotic tissue and flush it out with saline and hit it with antimicrobials, or oxygens will kill it too. So we have to be able to deliver it to the infection somehow. It's got to be reasonably priced. Like there's apparently there's a great cure for hepatitis C, but the medication is $90,000 to $100,000. So if you have Hep C and you're going to die, I guess that's the price you're going to pay for it. But it's not reasonably priced, but it is out there. Does not disrupt the host health by causing allergies or predisposing the host to other infections. All right. As I mentioned before, the allergies, I'm not going to say it's not going to happen immediately, but from what I can gather, you would have to have prior exposure to it, because you wouldn't even have antibody titers to it right away. It would take 5 to 10 days to even have titers to it, and you would have had the allergic reaction prior to that. So it's always the second exposure when you have that amnesic dose, and your body's saying, wait, I was exposed to this before, and now I'm going to have hypersensitivity to it. Now I'm going to be allergic to it. All right. So with chemotherapeutic drugs or any chemical used in the treatment, relief, or prophylactics of a disease. So prophylactics, just like condoms or other birth control or STD prevention devices. These are going to be used to diminish or prevent something from happening. All right, so the example of that might be you have endocarditis, you've got vegetation on your mitral valve or whatever, and you're going in for dental surgery. Dental surgery, you're going to be bleeding. We don't want the strep mutans from your teeth, or strep viridans from your teeth to get in your bloodstream and cause a reaction somewhere else. So prophylactically, we're going to give you some derivative of penicillin or cephalosporin or something that is effective on gram-positive bacteria, bacilli, or whatever, to diminish any adverse reaction we could have. Prophylaxis is use of a drug to prevent potential or infection of a person at risk. Antimicrobial chemotherapy is the use of chemotherapeutic drugs to control infection. All right, that should be relatively simple. And I don't mean to be too condescending to this, but when you read these, sometimes if you're nervous in an exam or whatever, you're like, well, what the hell are they asking me? So we're going to break it down for you a little bit. And if you get nervous, just take a deep breath and just read the word. And I'll be going over chunks of the word or what it means, or usually in the question, they'll give you some hint as to what they're asking for. And just remember, read them carefully. Nobody hates all but or not or whatever more than myself, but just kind of realize, if you're going through, well, that one is true, that one's true, that one I'm not sure, let me think about that, and break it down, and at least process of elimination for this, especially not so much for my exam, but NCLEX or board exams, you have to do that. All right, antibiotics, a term we're pretty all familiar with. Substances produced by the natural metabolic process of some microorganisms that can inhibit or destroy other microorganisms. So if you want to read your book, it'll tell you, how were these discovered? Well, they were discovered because, they saw mold growing on plates, and all around that mold was a zone of inhibition. They thought, well, these mold must be excreting some kind of enzyme diffusing into the plate that's inhibiting the growth of anything around it. And then back in the day, in the late 30s, early 40s, they thought, well, if I could excrete some of that maybe and purify it and inject it into somebody who has an infection, let's see what happens, right? And we could say that they were discovered in 1940s or whatever, but there's other documented proof that this stuff was going on in Egypt and China way before this. I don't want to say off-label, but it was done a little bit more surreptitiously. It wasn't quite as well documented or widely known. Semi-synthetic drugs are drugs that are chemically modified in the laboratory after being isolated from natural sources. So a lot of stuff we do now is semi-synthetic. So we're going to use the initial bacterium or mold or Clostridium strain or Streptomyces strain or whatever it is to get that. And we can grow that very, very quickly now. And then we're going to alter that side chain because we have to keep changing it up because the bacteria are finding ways to inhibit penicillinase or inhibit, like I said, the receptors, or if we can get it in there, they just produce more pumps to pump it out. It's kind of like our cells that can produce mitochondria on demand. Some of these bacteria are like, oh, well, this is not a good environment. We need more pumps. We need to pump this stuff out. It's kind of like bailing a ship. You have to, either you bail it out or you're going to go down with the ship. Now, synthetic drugs are antimicrobial compounds synthesized in a laboratory through chemical reactions. So those are purely synthetic, right? So they're not derived from any natural source. The chemical nature of it could be similar. So it could be like when they synthesize vitamins. I'm not a big fan of that because the chemical compound may be very, very similar. But when you consume that, it may not be bioavailable. Your body may not be able to absorb it. All right. And remember too, if it's missing any component of that synthetic vitamin or synthetic drug, if it's missing any component, it will literally grab it from some part of your body. So sometimes these things can actually make you sicker, specifically with vitamins too. You can take all these vitamins, and if they're missing something, they'll steal it from somewhere else. So that's a probability that sometimes these over-the-counter vitamins can actually make you sicker. All right, narrow spectrum. So I want to get into these two definitions. We always want to do narrow spectrum. It has much less broad effects, all right? And if any one of you are paying attention to the microbiome at all, and you should be, rather than watching Netflix or whatever, you might want to be learning about that. Antimicrobials effective against a limited array of microbial types. For example, a drug effective mainly on gram-positive bacteria. If it's going to start breaking down the NAG and NAM units of the peptidoglycan layer and cause a change in osmotic pressure, that's very, very narrow. And we want that because if we know something, the causative agent is gram-positive or very, very likely gram-positive, it's not a compound infection where it's more than one thing, we want to hit just gram-positive. We want to leave the gram-negative enteric of your microbiome alone. All right, or if we know it's gram-negative, we want to use something that's selectively more effective on gram-negative. We want to leave the gram-positive alone. All right, we really don't want to mess up the microbiome because if we start using these broad spectrum antimicrobials effective against a wide variety of microbial types, for example, a drug effective against both gram-positive and gram-negative, which is, what's been happening. These people have gone these great big long stints with broad spectrum antibiotics. They wipe out their entire microbiome, and the only thing that's left are these spore-forming bacteria, the Clostridium, and specifically Clostridium difficile. It's anaerobic. It likes to live in your large intestine because the oxygen concentration is very, very low. And once again, you wiped out everything in the lower GI tract and upper GI tract. Only what's left is Clostridium difficile because, once you wipe out all its vegetative cells, the endospore cells can regenerate, and then you have a population of just Clostridium difficile because there's no competition. And that is what's causing a lot of, primarily what's going on with these infections of C. diff. Right? So antibiotics are common metabolic products of aerobic bacteria and fungi or fungus. Bacteria in the genera Streptomyces and Bacillus. All right. Bacillus. And Streptomyces. Molds in general, penicillin and cephalosporium. So that's where we derivatives of penicillin and cephalosporin came from that. Right. Hope I didn't misspeak about the Clostridium before. If I did, okay. Yeah, so bacteria, Streptomyces and Bacillus, B. cereus and Bacillus anthracis. These are the ones that, so if I misspoke, we'll correct that now. So bacteria, Streptomyces and Bacillus, those species, and molds, penicillin and cephalosporins can make antimicrobials, right? So they're going to secrete a substance that's going to leach or diffuse out into their environment and inhibit any growth. Right? So by inhibiting the other microbes in the same habitat, antibiotic producers have less competition for nutrients in space. That's how they work. Right. So penicillium is from mold. We make penicillin from that. Cephalosporidium is mold. We make cephalosporins, and there's anything that starts with CEF or CEP or KEF. Those are all derivatives of cephalosporin. These are going to work on gram positive, and these are going to be the beta-lactam drugs, right? Micromonosporia, gentamicin, all right, generally, if I got that correctly, that is a protein inhibitor. Bacillus, all right, makes bacitracin and polymyxin. All right, polymyxins are going to be what's going to be working on cell membranes and things like that, polines. All right, Chromobacterium, all right, and Streptomyces. All right, so that's a filamentous bacteria, and I think your book has a couple pictures of that. And that's going to excrete chemicals, and these are streptomycin, erythromycin, tetracycline, vancomycin, amphotericin B. Now these are all work on the ribosomal unit. So whether it's the 50S, 30S, or the attachment site of P site or the A site, or maybe something can't attach at all, or maybe just the 30. Remember, the 30 subunit always shows up first. Then as the TMNR comes along, the 50 unit comes. So if we can screw up, jam up anything in that process, we can stop replication, and then the bacteria will eventually die. So here is penicillin, or penicillium, all right? And we can see here, all around here, is literally excreting something that's diffusing into this blood agar. Looks like blood agar. Yeah, if that's staph. Well, could be mannitol, but because the mold wouldn't grow on that, because mold likes a lower pH a little bit. So I'm going to say that is blood agar. Usually they tell you, but it looks probably like TSA or blood agar. All right. Interactions between drugs and microbes. Antimicrobial drugs should be selectively toxic. All right, remember that word's going to come up, and I did throw some questions using that terminology. Drugs should kill and inhibit microbial cells without simultaneously damaging host tissues. And to do that, like I said, we're going to really be narrowing in on the differences between eukaryotes and prokaryotes. And then we're going to come into a problem when we're dealing with, later on, we're dealing with molds, some of these funguses, a lot of these helminthes, and the protozoans, because they're eukaryotic. So some of the, like Flagyl and some of these other drugs will work on it, but we have to remember they're can cause damage to eukaryotic cells. And a lot of these protein inhibitors like tetracycline and things like that can have uh, drug effects because they also start affecting the ribosome of our mitochondria. So remember, the mitochondria of a prokaryote and eukaryote are different, except um, mitochondrial ribosomes are the same size as protozoan ribosomes. So if we're going to be wiping out the 50S or 30S or the 70S, well, 70S, so we're going to add them together. 1530s, 80, but we're going to call it 70. If you start affecting that, you can start affecting the mitochondria. And there's a legitimate thing out there called mitochondrial disease, where people literally they're not able to create enough mitochondria, or they damage a bunch of the mitochondria, and they get very, very I don't want to use the word anemic because that would indicate there's a hemoglobin issue, but they become very, very hypoxic, right? So diminished, not even that really. Not, you're getting oxygen to tissue, but you're not able to produce enough um, ATP. So let's just use the word lethargic. Okay. So as the characteristics of the infectious agent become more similar to the vertebrate host, complete selective toxicity becomes more difficult to achieve and more side effects are seen. So as we realize that our infective agent is more and more like our eukaryotic cells as the host, or humans, we start seeing some of the toxic effects become more prevalent, right? So, like I said, I'm not going to ask you guys specific drugs, unless I asked you about super popular like penicillin, cephalosporin. I'm not going to ask you about vancomycin or some of these bacitracin. Some instructors might. I don't really feel it's important, but I would expect you to know the class of drug, or where did it come from. So, like, just say if I said, what is the class of drugs of tetracycline? Or, what's its mode of action? Well, all you have to know is it's gonna affect the ribosome. So it's gonna be some form of protein synthesis inhibition, right? So I'm not going to be specific. Well, does it work on the 50S or the 30S? Does it work on the A site, amino acid site, or the protein site, or polypeptide site, or the exit site, the E site? That that's pretty picky. Some instructors might. I'm not going to say you're never going to see it, but I wouldn't ask you that. But I definitely want you guys and girls, to know the basic, there's 5 of them, modes. How do they work? And then remember too, if you have something that's very, very resistant, they might give you something that works in 2 or 3 different modes of action. So we can definitely wipe it out if it's very, very resistant. All right. At the end, we're going to talk about how this resistance developed, and how, as healthcare providers, we can hopefully change that, because at some point in time, there's some staff out there now that we just cannot get rid of. That's, we'll get into those reasons in a bit here. All right, so I'm going to say, if I had to pick a majority of the classes that right now, a lot of these are going to be cell wall inhibitors. So it's going to be your beta lactams. So penicillin, cephalosporin, vancomycin, bacitracin. These are the 3 that you'll see, and they've been around forever. So they're going to work on the cell wall inhibitors, and they break down the protein linkages between, if they're NAM and NAG units, the protein linkages that connect all the peptidoglycan layers. And remember that these gram-positive can have, literally from 30 to 80 to 100 layers of this. If we can go in and break down the protein linkages, it's kind of like I mentioned before, like chiseling out all of the mortar between your cinder blocks in your basement. So when it rains or whatever, water just comes in, right? Because we've broken down that barrier wall, and that's how these work. So the osmotic pressure outside of gram-positive bacteria is generally always higher. So water wants to come in, but that cell wall is stopping it. So if we can alter that cell wall, we can cause the fluid or osmotic pressure to come in, and that cell will just blow up. It'll just burst. Lyse, if you want to use that word. All right, cell membrane. Now this works, all right, but we generally don't want to just be indiscriminately prescribing this or distributing this or dispensing this, because remember, we have a biphospholipid layer. Now the cholesterol or the structural units, we have sterols and cholesterol. These other organisms are estradiol, and they're going to have slightly different. But depending on what these bind to, they cause loss of selective permeability. So these are how the polymyxins work. And remember, your biphospholipid layer is what determines what comes in and out of your cell. Same cells of your body as a eukaryote, or the bacterium as a prokaryote. So if we can go in and we kind of, I think we'll see a picture of this, the biphospholipid layer, the lipid tails are in the middle, the phospholipids or phosphates on the outside, facing the water, either outside the cell or inside the cell. If we can go in and we can start moving that apart, all right, we can cause gaps and things can move back and forth. So if this is a normal cell, only something that is theoretically has close to a zero charge, it's lipid soluble, or it's very, very tiny, can move back and forth. But if we can tear holes in that and let great big particles in, or cause fluid to leave, that cell will either crenate, dry up, or lyse if water was rushing in. Now, DNA or RNA. So we're going to go in, and remember, this is the replication. This isn't the transfer RNA making the proteins. This is literally the replication. So whether it's, directly from RNA or making RNA from DNA, this is how they're going to work. So inhibit replication and transcription. They inhibit gyrase. Remember, gyrase is what's going to go in and attach and unwind that, and we put this SSS, the single soluble proteins, on there to hold. We unzip it. We want to hold that zipper open. This inhibits gyrase, so it's not able to go and attach and unwind, so we can't even replicate. All right, so this is how the quinolones, ciprofloxacin works. All right, and Cipro is huge, is very, very effective. All right, but what we don't want to do is, when we have these drugs that are super effective, we don't want to be indiscriminately dispensing these. We don't want bacteria to figure out a way to get around that. All right. So even if we have something out there that can get around it, it may have a mutation to do that. But unless we expose it to Cipro or ciprofloxacin, that's not going to be expressed. So we really don't want to use it if we don't have to. All right. And then we can inhibit RNA polymerase. All right. Rifampin does that. It works in inhibiting the polymerases coming along. And I think we read that they were using rifampin for people that were in a coma, and for some reason, I think that's the right drug. There's a research where they were coming out of the coma for like a half an hour. It was the craziest thing. And I was talking to a couple of the other doctors at school, and I'm like, well, we couldn't figure out, how that would happen. And then I forgot about it. But anyway, trying to figure out the mode of action for that. Anyway, all right, so number 4 is protein synthesis inhibitors acting on ribosomes. So there's a lot that of these. And also remember too that whether it's gram positive or gram negative, they all have to reproduce proteins, right? And they all have to be able to replicate they have to be able to replicate RNA or DNA. They have to get the information to make the proteins, right? So if we can stop this or this is gram positive or gram negative. These are very, very broad spectrum. All right, and we'll see coming up like how some of these work, and you guys might think it's boring, but it's super cool that if I can stop this transfer RNA that has the amino acid attached to it, if I can stop it from floating around and coming here because of the codon and nucleotides, that's super cool. If I can bend this 30S ribosome so it doesn't attach, that's great. Or if I can go in like tetracycline go in and just block this, it's like, no, not going to happen. All right. So, as you get in the field, just start learning how these all work, maybe, if that's important or not. And then coming up, we're going to see tetracycline was extremely popular when I was a child. So, early, mid, late 60s, very, very popular. Work, broad spectrum, you had anything, would wipe it out. The problem is it's absorbing the tissue. So you'll see people my age, so 50 to 70 or 80, they're going to have very, very brown teeth. And it's not because they never brush their teeth. It's because the tetracycline, the medication, got absorbed into their calcium, into their teeth and their bones, right? So it would cause some major, major problems. All right, and the last one is metabolic pathways and products. This is going to work. These are how the sulfur drugs work, and they're going to work on PABA, folic acid, which you need to produce purines and pyrimidines. So we need it for DNA replication. And I always mention, no matter what course I'm teaching, remember that women of childbearing years, you need folic acid B9 and B12. So you might as well take them all together. And what the hell, you might as well take a complete complex B vitamin supplement. Because if you have a lack of folate or B12, can cause some major, major birth defects. All right. So blocks the pathways and inhibits metabolism. Sulfidamides, the sulfa drugs, trimethoprim. This actually works in the second stage of that metabolic pathway. So it blocks the production of folic acid, and these organisms can't produce the purines and pyrimidines. All right. And then we're going to see coming up that what they'll do is they'll just find another pathway. All right. They become resistant to it. They'll just find another way. All right. So just showing you here again, inhibition of cell wall synthesis. So, and this really works much better on younger growing cells. Same with when I was talking to you guys earlier. I made the video, I wasn't talking to you, but on the video I produced about the staining. The young cells always stain better than older cells. So I like to stain something, if it's under 48 hours old. All right. Inhibition of protein synthesis. All right. So once again, these are going to work on the ribosomes. All right. And they can make antisense nucleic acids. They can give you an analog. They'll give you like a fake nucleotide, like a fake thymine or a fake adenine or guanine. It's super cool. They just put a fake one in there. And then when you go to reproduce that, the codon doesn't work. It's super cool. And then disruption of cytoplasmic membrane. Remember, this is the polymyxins. All right, for bacteria. And then the polynes we use for the antifungals. But remember, with these polynes can start affecting our biphospholipid layers too. The antifungals have estradiol instead of cholesterol and sterols, but it still can cause some major problems. And a lot of these polynes or some of these antifungals or nystatin can only be used outside the body. And some of these drugs that they take for, like, let's say you had some kind of a toe fungus, these can become pretty toxic. All right. So inhibition of general metabolic pathways. Once again, this is fulminamides. Trimethoprim works on that second stage between PABA and folic acid. There's a tetrahydric acid. It can go in, it starts working on that. And what will happen is these, if they become resistant, they'll just find another pathway. Kind of like if you were in Buffalo and you're driving by the Galleria Mall or whatever, and you see all that traffic. Well, you just get off at the first exit and you just drive around the city the other way. You'll get there quicker, less aggravation. They'll just find another pathway. Inhibit inhibition of DNA and RNA synthesis. Remember, this is actually how they're going to block that polymerase or gyrase or helicase and the polymerase. All right. So these actomycin nucleotide analogs, right, they give you that fake um, nucleotide. All right. This is how rifampin works and the quinolones, right, how they work. All right, and this really is some of the antivirals work. Remember, the viral cells, they have to find one of our cells to live in. They're intercellular parasites, right? So we don't want to kill the cells. I mean, we will if we have to, but we can stop the attachment site. If we can stop the attachment site, they can't get in. And then once they get in, we can mess around with their ability to produce replicate the RNA or get into the nucleus for DNA transcription. And then if that doesn't work, we can hit them with like a protease inhibitor on the way out, so they can't to reassemble their proteins. Right, so we have ways around it. All right, that was a video that didn't work. All right, so here are some of the bacteria, funguses, protozoan, worms, and viruses, how these will work. I'm going to hold off on these a little bit. Wouldn't hurt to just briefly read through these and figure out. But like I said before, with the viruses, general targets, cell membrane, we might make sure it doesn't attach. DNA synthesis, we can stop it from synthesizing, and then reverse transcriptase. When it goes to leave, it won't be able to assemble. Yeah, blocks DNA formation, stops the virus from replicating, and blocks the entry. So those are the three modes of action for that. There's a fourth, but I'm not really worried about it. Helminthes, worms. So we can inhibit their metabolism if they starve. We can kill the larvae. We can stop their attachment site like the Scolex. We can actually paralyze their muscles so they're unable to move, and then we excrete them. Protozoan, nutrition of malarial parasite. I had some, they gave me some of this before I went to India. Yeah, so it's going to build up toxins ways. We just kind of kill the cell by not allowing it to, force it to live in a dirty environment. All right. All right. Hopefully this will play for you guys. But this, listen, you guys all have mastering. All right, so go in and watch these. Sometimes the information is more than you need to know, but just at least watch the animation. All right. So as far as I'm concerned, these are probably some of the best that I've seen. Or you all have YouTube, so something you don't quite understand, you can go on YouTube it, find anyone better than me explaining this to you. All right. So the spectrum of drugs. So narrow or broad spectrum, just know the difference. Right? Remember, narrow is going to effective on a small range of microbes, target a specific cell component that is found only in certain microbes. All right, so if it's a gram negative, it's going to be, it has that double cell wall, it has those porins, it has that periplasmic space. It can excrete LPS. So we're going to, anything we can use to defeat it, we're going to, right? Broad spectrum, or, if it's gram positive too. If we know it's gram positive, we can just hit it with a cell wall. Use a cell wall inhibitor. So that is the major reason behind the gram stain. So why don't you do gram stain? Well, number one, it's cool to do. And number two, way back in the day when we didn't know quite as much, at least we could figure out, even if we did a culture and sensitivity, which they don't even do anymore, you could say, well, I have an infection, or my kid has this sore throat. Well, we probably pretty much know what it is, or have UTI or whatever. We would do a culture and sensitivity. We're going to go in and grow it and then stain it. And we can say, by the next day, oh, well, your causative agent here is, we'll look at it and just say, it looks like it's gram-negative rods. Looks like it's E. coli, or, these are purple clusters, so we know it's some kind of staph. At least we know what antimicrobial to use rather than using the broad spectrum. Once again, that wipes out gram-positive or negative, and specifically, we don't want to wipe out the microbiome. All right, so I'm going to go through just the classes and some general stuff, how they work, go through the 5 classes here, give you an idea of some of the drugs. I'm not going to get really in depth about that. It's really more for your pharmacology courses, but at least you have in the back of your mind what the modes of action. All right, most bacterial cell walls contain peptidoglycan, that's sugar. All right, so remember, in a gram-positive, it's the outer layer, it's super thick, and the gram-negative, it's kind of that middle layer between the 2 cell walls. They both have it, but if it's gram-positive, it has that 30 to 100 layers, and if we can break down the protein units, the NAG and NAM units, we can change the osmotic pressure. So penicillins and cephalosporins block synthesis of peptidoglycan, causing the cell wall to lyse. All right, so we just kind of chisel away at the mortar. Active on young growing cells, penicillins that do not interfere with the outer membrane, penicillins that do not penetrate the outer membrane are less effective against gram-negative bacteria. Because remember, they have that outer membrane with those porins. If we can't penetrate the outer membrane, we're never going to get to the peptidoglycan layer, so they're pretty much pointless. All right, but then we have, we can have some semi-synthetic drugs that can get us, they'll figure out a way to get us through that cell wall, outer cell wall, and into that middle peptidoglycan layer, and those be a little bit more effective. Right. Broad spectrum penicillins and cephalosporins can cross the cell walls of gram-negative bacteria, and we just kind of figured that out and give it as a broad spectrum if we had to. So here's just kind of showing you, these are this is the NAG and NAM units. But we're going to break up these cross bridges. All right, so the cross bridges, we break it up so these can't, and they can't hold fast. Right. So antimicrobial drugs that disrupt the cell membrane function. So these are going to be your polymyxins. All right. Amphotericin B and nystatin. Some of the, like nystatin, I'm pretty sure you can only use on the surface. But a cell with a damaged membrane dies from disruption and metabolism or lysis, just like our biphospholipid layer. These drugs have specificity for a particular microbial group based on differences in types of lipids in their cell membrane. So we're going to really target the types, we have biphospholipid layers. So if we can have one that targets very, very specific lipids, go in and we can disrupt that. Polymyxins interact with the phospholipids and cause leakage, particularly in gram-negative bacteria. Right. And just kind of keep in the back of your mind, well, wait a minute, we have phospholipids in our cell membranes too. So amphotericin B and nystatin form complexes with sterols on fungal membranes, which cause leakage. So it's kind of cool to know that now, but when we start talking about molds and stuff later on, these are going to be, amphotericin B is going to be pretty much the gold standard. And nystatin, usually that's going to be used externally, and depending on the dosage or the concentration of that. So polymyxins just go in and they just disrupt that cell membrane. So they're going to work on different types of lipids, disrupting that. Drugs that affect nucleic acid synthesis. So remember, is this the ribosome, or is this DNA or RNA replication? All right, so these are nucleic acid synthesis. All right, so ribone, nucleic acid, DNA, or deoxyribonucleic acid. So just nucleic acid, just think, oh wait, that's got to be DNA or RNA. May block synthesis of nucleotides. Those are those purines or pyrimidines. All right, so if we can't produce them, right? Inhibit replication or stop transcription, right? So we can stop the whole replication if we can stop that helicase from unwinding it, or if we can stop the single soluble protein, or scared the crap out of me. All right, chloroquines binds the crosslinks to the double helix. Okay, so those can really work on those solubility proteins. Quinolones inhibit the DNA helicase, and you're going to see there's going to be all kinds of derivatives. So just look for the ions. They might change the beginning, or put ly inolones. They'll just change that. So kind of look if you're doing taking a pharmacology exam and they say, well, what would the drug so and so? How would it? What's its mode of action? Just go by the derivative. What chunk of that did they use? Right. Antiviral drugs are analogs of purines and pyrimidines. Insert in viral nucleic acid preventing replication. So antivirals, remember, they're going to go in, they're going to give it a fake purine or pyrimidine. So they're going to give it a fake thymine or a fake adenine or a fake cytosine or a fake guanine. So when it goes in, it looks similar to it, so it binds, but then when you go to replicate it, it can't replicate it. Right? It can't replicate it accurately. So drugs that block protein synthesis, remember, whenever you said from day one, I said, whenever you think of ribosome, especially rough ER, right, rough endoplasmic reticulum, that's ribosomal. That's always going to be amino acids put together to make polypeptides, and polypeptides turn into proteins. And as the proteins leave, those R groups, depending on whether they're water loving, water hating, negative charge, positive charge, or whatever, they're going to twist and consort. So whether they're going to be attracted to each other or repel each other, that's how these proteins, when they leave, they go into primary, secondary, tertiary, or quaternary shape. All right, and then remember again, when you start changing the environment, pH, temperature, whatever, it starts denaturing, it starts breaking those bonds. So these things can start unraveling. And then remember, once a protein form follows function, so once a protein changes shape, it's no longer functioning. All right. So ribosomes in eukaryotes differ in size and structure from prokaryotes. Antimicrobials usually have a selective action against prokaryotes. Can also damage the eukaryotic mitochondria. And I put that in red. So we're going to target its differences, but I want you guys to always remember the only exception is our mitochondria, the ribosomes in our mitochondria, which, by the way, your mitochondrial DNA you got exclusively from your mother. All right. That had nothing to do with it. That can be damaged. Aminoglycosides. So amino, protein, right? Glyco, sugar. All right. So streptomycin, gentamicin insert on sites of the 30S subunit and cause misreading of the messenger RNA. So if it gets stuck, aminoglycine, okay, that's, oh shoot, that is, all right, protein to sugar, okay, that's got to be protein synthesis. I know that happens at the ribosome, so at least you've gotten that far, right? This just happens to work on the 30S. Remember, the 30S, when the messenger RNA is coming through, the 30S goes, oh, wait, I have a mission. It dots onto that. And if attachment's right and the nucleotides line up and the codon lines up, then the 50S comes along, drops in, and then we run that ticker tape through and we start asserting the amino acids, and they click on to the next one, and that polypeptide gets bigger and bigger and bigger. Then it hits that stop codon, AUG, and then it ejects. It's like it cuts it, and then it leaves that site there. All right, tetracycline, block attachment of the transfer RNA on the A acceptor site and stop further synthesis. So this literally, tetracycline comes in and attaches to the messenger RNA, and it blocks that site. So now the transfer RNA that's coming along with the amino acid on it can't attach. It got blocked. All right. All right. So, the aminoglycosides, I said, remember, you're going to work on your ribosomes. These just happen to work on the 30S. All right. All right. And then some of these other quinoxalones, just showing you how they work. Not that I would test you on it, like tetracycline right there. It just blocks that. It can't even start. Erythromycin is prevented from translocating, so it blocks it here, so it can't even leave. Not that it's important to remember that. I just think it's kind of cool that how these different drugs can work differently. All right, so sulfonamides, remember these will make the PABA, and I said that trimethoprim works in the second stage of this, the tetrahydrofolic acid. So these are going to block PABA, which is a precursor for folic acid. And remember, folic acid right here, it says, what your body needs. Your body needs it too. Same bacteria to make the purines and pyrimidines. And remember, the purines and pyrimidines are those adenine, thymine, cytosol, guanine. So those are those nucleotides. So depending on whether they have 2 bonds or 3 bonds, remember the DNA helix, the hydrogens were in the middle, then we had the sugars, and then the phosphates were on the end, and then you had a 5N, a 3N, an H on one end, OH on the other. All right, so these are going to go in and block that. All right, so they're gonna look. This is sulfa drug, looks just like this. It goes in and blocks that binding site. Right, so it blocks the receptor. All right, so what's going to happen is these drugs are either going to be able to change their receptor site shape, which doesn't usually happen, or they're just going to say, you know what, screw it, I'm going to just go around this whole step. I'm going to go from here to here. I'll find another way. All right, so there's the structural differences. There's PABA and there's the antimicrobial. All right, so they look very, very similar. So if this goes in and binds up all the sites, PABA can't get there, and then they can't run the enzymatic reaction to produce the folic acid. All right, and then synergistic effect. Just remember, if you have something really stubborn, we'll just do like 2 antimicrobials or 3. So 1 and 1 is like 3, and then if you do even more, so the more antimicrobials you do, the more synergistic effect it has. So antibacterial drugs are like your antibiotics or synthetic drugs. These would be derivatives of the natural, all right, from the bacteria or the mold, or synthetic. We're going to go in and change that side chain. All right. So your antifungal drugs, these are going to work on the fungus. Remember, these are going to be like your polymixes, your amphotericin B, nystatin, things like that. Antiprotozoal drugs like Plaxil go in and they kill those. So if you were, going to a foreign country and you thought maybe you'd be exposed to some protozoan from consuming water or whatever, you can take those. And then antiviral drugs, whether it's for herpes or whatever, we can change the receptors maybe. We can change the bonding sites on the cells. Once it gets in, we can stop it from replicating the information. We can maybe stop it from getting into the nucleus, or if it gets past all those on the way out, like I said, reverse transcriptase, we can go in and we can make sure it doesn't replicate properly. And the other thing I want you guys to remember too, viruses can only attach to very specific cells. They have to have very specific receptor sites. Remember that all of your cells have the HLA-1 markers that tell your cells that they're yours. It's a little bit more complicated than that, but for right now, we'll go with that. But these viral cells can only get into certain, like hepatitis. They can only get into liver cells, or or HIV can only get into antigen-presenting cells or CD4 cells, right? Coronavirus can only get into cells that have an HCE, um, antigen-converting enzyme marker. So that, is your pneumocytes, kidney, the liver has some too. So when we're seeing a lot of these different effects of corona, it's because these are attaching to these cells in these areas that have the ACE marker on them. Right. So beta lactam, I mentioned earlier, this is going to be most of your penicillins or cephalosporins. All right. And they go in and they break up those cell walls in the gram positive. So just kind of keep that in mind. Primary mode of action is to interfere with cell wall synthesis. Greater than half of all antimicrobial drugs are beta lactams. And remember, these are going to be derivatives, CAF, CEF, sporins. So just kind of look for, they'll change the beginning of the word or whatever. They could be generic, or they could be second, third, fourth, fifth generation of the same thing. Penicillin and cephalosporins are the most prominent. Right. Okay, so really all that you need to get from this is, here is the beta lactam. All right. Diazolidine and the beta lactam. All right. These are your side groups. This is what makes each medication different. All right. How they spell it different or whatever. But this is the major component that's very, very similar. All right. That's a mode of action right there. All right. Penicillin G and V are most important natural forms. Penicillin is a drug of choice for gram positive because it works on that gram positive cell wall. Streptococci. So if you had like let's say you had a tooth infection or whatever, they're going to be 99.5% sure it's going to be some kind of strep, viridans, strep mutans. So we're going to give you some kind of penicillin drug for that. Or if you have, meningitis or syphilis, the spirochete likes to burrow in your mucous membranes, but also inhibits IgA antibodies. They're going to give you some form of penicillin G probably. Semi-synthetic penicillins, ampicillin, amoxicillin, they're broader spectrum gram-negative infections. So we said that some of these have a way they can get them into that gram-negative through that cell wall, through the porin. So they can work in the peptidoglycan layer of gram-positive and the gram-negative. All right, and then we have the major problem right now. We have penicillinase resistant. So these drugs are able to either change their bonding site, or they can actually go in and they can have more efflux pumps. So we can maybe get the medication in, they just pump it right out. And the problem right now is we have methicillin, vancomycin, and methicillin resistant Staph aureus. So no matter what we give them, it's resistant. We don't want to use Cipro. And the other thing that we have to do is we have to hit it with more than one antimicrobial. But the problem is if they become resistant to that, then we're in serious trouble. Primary problems are allergies and resistant strains of bacteria. And once again, the allergy problem is if you took this and didn't make sure it was all completely gone, and there was some residual, and you reinfected yourself, and then you took the medication again, you could have allergic reaction because you would have built the antibodies to it the first time, because these medications are foreign to you, right? So your body should be building antibodies to it. But if you have a hypersensitive reaction to it, it can cause major problems the second time you take it, right? And we'll talk about taking antimicrobials the full time to make sure it's wiped out. Because remember, when you're doing this, it's going to be survival of the fittest. It's going to wipe out the weakest bacteria first. It's not going to wipe out the strongest. So you want to make sure you take it to full course. Okay. And just realize, I want you guys to know the difference between narrow and broad. All right, because broad will work on both. We have a way of getting it into the gram negative. And either way, these are all going to be cell wall inhibitors. So gram-positive has the thicker outer wall. It's a little bit easier to get into. But if you have that, I'm sorry, that has a thicker peptidoglycan layer, easier to get to. The gram-negative has that cell wall we have to get through first before we get into that middle layer. Right? So that's why we need these broad spectrum. Right? It works on gram-negative by getting through that cell wall. Right? And there's some, some disadvantages here. Cephalosporins, these are going to be cell wall inhibitors. They account for 1/3 of all antibiotics distributed synthetically altered beta-lactam structure. So if we had something that was penicillinase resistant or resistant to penicillinase and caused fewer reactions, so they worked for a while. And this is where I said they can start with Ceph, Caf, or whatever. All right. All right, and then we also have to worry about, if we're taking some of these orally, what is the stomach acid, or what's the digestive system doing to it? Is it breaking it down before it gets into the bloodstream? And then polymyxins, these are the ones that work on the cell membranes. Narrow spectrum peptide antibiotics with a unique fatty acid composition, because that's how they get into the cell membrane. They have a very unique fatty acid composition, and that's how I said with these cell membranes, you can only get into these if you are lipid soluble, zero charge, have to be really, really tiny. All right. So these treat resistant Pseudomonas and severe UTIs, and usually these are gram negative. All right. Fluoroquinols, and I put this in red. So they're going to chunk that out, right? As they change the name of it, this is going to be a component of it. Works by binding to DNA gyrase. Remember, if it binds to the gyrase, it can't unwind, so you can't replicate the genetic information. So it's broad spectrum of effectiveness. It works on everything because everything has to be able to replicate its genetic information to make the proteins or make the enzymes. It has to get the information from somewhere. Right. Concerns have arisen regarding the overuse of quinoline. This is the part I put red there. Right. Drugs, because CDC is recommending careful monitoring of their use to prevent ciprofloxacin resistant bacteria. So if we this will work, but if we give it to everyone and we have a resistance to it, then we have nothing in the arsenal, right? And, it may not be a concern to you or I, but if someone's on the front lines or somebody is triaging an outbreak or whatever, we need to be able to have something for them. Aminoglycosides, remember I said that these are, think of protein, this is going to be the 30S unit usually, all right, composed of one or more amino sugars, and the aminocytolidol rings binds ribosomal subunits, and I'm pretty sure this was the 30S. Not that that's important. All right, products of various species of soil, Actinomyces, and the genera Streptomyces, right? So that's where we got this from. It's pretty cool that we were able to find something in nature and then exploit it. Not the word exploit. We were able to take advantage of it. Let's put it that way. So broad spectrum inhibit protein synthesis and gram-positive or gram-negative, especially useful against aerobic gram-negative rods and certain gram-positive bacteria. And this is what they'll use. Streptomycin, we've all heard of this, for bubonic plague, malaria, and TB. All right. And I said before, I'm a little concerned about what's going on right now in March of 2020 and now specifically March of 2021. If people want to get vaccinated, they can, that's fine. I would never say not to do it. It's a personal choice. But if there was an outbreak of bubonic plague from fleas or malaria is usually in rodent urine, I don't think I'm going to be coming in contact with, or tuberculosis, which is, I predict is going to be a little bit more rampant in about 2 years, because we have some incoming people under the country right now that may be carrying this. All right. I would be vaccinated against this, but we need these medications to counteract these. These are some serious diseases. In my opinion, a little bit more serious than the flu, but that's just my humble opinion. Gentamicin, we've probably all heard of this. It's less toxic, used against gram-negative rods, so they could use that against a UTI, E. coli, or whatever. Yeah, there's just some other ones, and I won't even hold anybody accountable for it, and I personally don't care. I'm not prescribing medication. That's more of a pharmacology thing, or when you get into practice. Right. Tetracycline, this is very common. I have seen several questions on tetracycline and why it isn't used as much as it used to, and they just remember it has some deleterious effects. We'll see a picture of someone's teeth coming up here. I know 4 or 5 people who don't have white teeth, and you can't even bleach them or whatever, other than crowns or something. You can't get your teeth white again because it's this deep into the tooth dentin. Right. So broad spectrum blocks protein synthesis by binding to the ribosomes. Remember, tetracycline, not remember, but tetracycline bonds right at the 30 sub, the MRA comes in, and then as the 50 comes down, it jams it. It's not able to attach. So this is a treatment for STDs, Rocky Mountain spotted fever, Lyme's disease, tetanus, acne, and protozoa. So protozoa have to reproduce their proteins too. So yeah, here's the thing: if you have certain STDs, Rocky Mountain spotted fever, which is causing intravascular coagulation of your bloodstream from that LPS released from the gram-negative bacteria, Lyme's disease, typhus, or severe acne, protozoa, then maybe this is what we want to go with. All right, generic tetracycline is low in cost but limited by its side effects. All right, so all here is you get that black tongue, very attractive, and then the brown teeth, so they can definitely diminish your social activities. Okay. Erythromycin, we probably all heard of this. So this is going to be attached to the 50S unit. So tetracycline, if they ever ask you, attaches to the 30. Erythromycin attaches to the bigger unit. Remember, kind of 30 always comes in first after the messenger RNA. And like I said, some of these can bind to the A site, P site, E site, whatever. That's really picky. I don't know if that's extremely important, but if they ever ask you, at least you've heard it. Take it orally for mycoplasma pneumonia, Legionnaire's disease, chlamydia, pertussis, very, very problematic in children, diphtheria, and prophylactic prior to intestinal surgery. If somebody's going in for a fistula or something, they have a recurring IBS or something, they can give them to prophylactically, right, so that microbiome, if anything in there, doesn't get into the bloodstream and things like that. For penicillin-resistant gonorrhea, syphilis, and acne, right? Drugs that block the metabolic pathways. So just remember, these are all the sulfa drugs. Narrow spectrum, they block the synthesis of folic acid by bacteria. Right? And primethylone gives the combination. All right, so primethylone, that works in the second portion of that acid synthesis. Anything that ends in the azoles too. All right. Ketolides. All right, there's a new drug with different ring structure than erythromycin. And these are just showing you that they can go in and they're going to just hijack the structure of this. They're going to make them slightly different. As things come up, we can just go in and change it, and it may buy us a little time. Yeah, VRE, or vancomycin-resistant enterococcus. All right. All right, and pollens are used primarily for fungus because I said they work. Fungus has very similar or more similar cell membrane to us than bacteria. So we want to go in and find something. And these are amphoteric, all right? So they're water and fat loving. They can go either way depending on which end is used. So fungal cells are eukaryotic. A drug that is toxic to fungal cells is also toxic to human cells because they're both eukaryotic. So 5 antifungal drug groups, the microglider polyene, this is amphotericin B. This is going to come up over and over again whenever we're talking about fungal treatments. So it's going to, amphotericin B is usually always the answer they're looking for. So it mimics lipids, so it's going to be able to be soluble, right? Most versatile and effective, topical and systemic treatments. And remember, topical is going to be put on the skin, usually in a concentration that you wouldn't want to be taking orally, and they're amphoteric. All right, nystatin, you're going to see that it's topical treatment only. You do not want to ingest that. And I think, if I remember correctly, nystatin is in a lot of can be in a lot of fungal creams, athlete's foot, jock itch, stuff like that. I want to say yeast vaginal infections, triple antibiotic ointments. Sometimes we'll put nystatin in there in case, like, let's say you got cut or you want to hit it with. If you look at your triple antibiotic ointments, so if pull it out, take a look at it, and you'll see they're going to be antibiotic classes. Whatever's in there is going to be from, like, one might be a cell wall inhibitor, one might be an RNA inhibitor, one might be a ribosomal inhibitor. So it's kind of cool. They take 3 different modes of action of 3 different drugs and put it together in an ointment. So when you cut yourself and you want to put that ointment on your skin, you're covering all your bases. All right. And I don't really have a problem with that because the cut's immediate. You want to kill that organism before it gets into the system and becomes systemic. All right. So I think it's a super cool idea. All right. 5 antifungal drug groups. All right. You've probably seen this before. And then amphotericin B and any of the azoles are generally always what they're going to be asking you for. So when you see, like, these all end in azole. All right. So when they ask you, if they give you a case study and you're thinking, oh crap, this guy, it sounds like he's got a fungal infection. All right, so it's going to be nystatin, polymyxin, or an azole is going to be the answer. All right, so antiparasitic chemotherapy, and we'll talk about this when we start talking about protozoa, things like that. So antimalarial drugs, quinine, all right, still used. It's from tree bark, all right? They still really haven't found anything better than that. So I put that in purple here. Metronidazole is what they gave me before I went to India in case I consumed anything. Tetracycline still works. Happy to say I went to India, no issues whatsoever. I didn't get malaria. I didn't get sick. Eating with my hands was a little weird on banana leaves, but it's okay. All right, hemolytic helminthic drugs. So helminthics are worms. So these immobilize, paralyze them, so you shed them out when you defecate, disintegrate or inhibit metabolism, so we can starve them out. They can't. Once again, these are going to end in AZOL. Right? Yeah, glycosamide, I've seen that. That actually just destroys the skolk of a tapeworm. So if you really want to lose some weight, you always tell students, you want to go online and get a tapeworm. Please don't do this. But if you do get a tapeworm, you got to figure out whether it's going to be the pork tapeworm or the beef tapeworm. All right. Because the beef tapeworm can't leave your digestive system. The pork one can. So if you're going to get tapeworm, try to make sure you get the beef tapeworm. And then, when you lose the amount of weight you want to lose, then you get a little medication like this. It destroys the scolex, and then the tapeworm has to let go. All right. And then what you want to do is you want to grab that feces for the next couple of days. And you want to put gloves on and a mask. You want to sort through that with your fingers. It's a shitty job, but someone has to do it. And then you want to find that scolex. And then once the scolex is going to go, okay, tapeworm's completely gone. It can't come back because in the meantime, it's been dropping off segments. And you're flushing it into the sewer system, which is awesome. But just a side note there. All right. Okay, great. Thanks. All right. Antiviral chemotherapeutic agents. So antivirals, remember they're only in specific cells. Viruses are intracellular parasites. So we can't go in and just kill all those cells. I mean, we could, but what's another way around it? So can we force it to release chemicals to warn everyone around it? Warn everyone around it. Hey, listen, if you come across this viral guy, then and somehow gets inside you, I want you to kill yourself. I want you to go through apoptosis, so you're not a virus-producing factory, right? Or selective toxicity is almost impossible due to the obligate intracellular parasitic nature of viruses. So what we want to do is we want to block the penetration into the host cell. We want to be able to make sure that this virus can't dock, can't find a receptor. If it finds a receptor, we don't want the cell to let it in through endocytosis, right? Or once it gets in, can we block the replication, transcription, or translation of the viral genetic material? So depending on whether it's a retrovirus or a DNA virus, we want to be able to block, able to make a duplicate copy of the genetic information, or we don't want to hijack our ribosomes or DNA to use us as a duplicating machine for them, right? So the cyclovir for the herpes, and my friend Joe used to call him the herpes guy. He was a rep for that. And then there's ribavin, which is a guanine analog. So that's kind of cool because guanine, attached to cytosine. So, but if we can give it a fake guanine, so when it binds to cytosol, that it makes this totally erroneous code, and then the virus can't replicate. It's super cool. All right, and AZT, you've all heard of this, right? For HIV, it's a thymine analog. All right, so like I said, for viruses, we can just go in and make a bunch of analogs. We can make a bunch of fake nucleotides, right? And then, once it gets in, we can stop it that way, or prevent maturing of the viral particles. So if they get past all of this, then on the way out, we give them protease inhibitors, right? So if you were taking a HIV cocktail years ago, we give you a little AZT. If HIV was able to get into your CD4 cell or your monocytes, any of your dendritic antigen presenting cells of any type, they can get in. We can go in and give it an analog. We can give it a fake nucleotide, so it's going to replicate erroneous information. And then if it got past all that, on the way out, we can say, we're going to give you also a little protease inhibitor. So on the way out, we're going to make sure it garbles everything up or it can't assemble it right. So even if it ejects viral particles, they're defective. All right, totally cool. All right. Rimantadine restrict almost exclusively to influenza A viral infections, prevent fusion of the virus with the cell membrane. So we can just say here's a drug. I don't care about the drug classes, but just realize first thing we do is we make sure it can't attach. All right. Number two, we can opsonize or neutralize the cell receptors. We can say, you guys can float around all you want, but we've changed the dock. You can't, if it's, let's just say it was a coronavirus, it's not, but we're going to say that component won't fit in here because we've covered the site. We didn't alter the shape, we just covered it. We put some super glue in the lock. Let's put it that way, all right? Number three, we can block the entry of influenza virus by interfering with the fusion of the virus in the cell membrane. All right. So, okay, you dock, that's great, but now, you know, this virus, once it docks, the cell has to go and engulf it, come together and bring it in. And if it's encapsulated, it actually is so nice. It actually takes the capsule off and hangs it up in the coat room. And then if it's a DNA virus, it says, oh, here, by the way, the nucleus is right there. Have a little snack, and then go help yourself to the nuclear machinery in there. All right. Number four, stops the action of the influenza remedies required for the entry of virus into the cells. We can go in and we can change the ability for that to get in, or actually once it reassembles, we make sure it can't leave properly. Right. Right. And here's a cycle things for herpes, just showing you how it works. All right. Oil and topical treatments for oral and genital herpes, chickenpox, and shingles. All right. So these are Valtrex. I'm sure you've all seen commercials on that. So if somebody has herpes, whatever, you put a little Valtrex on that, and it will have it heal up faster. Right. Retrovirus offers two targets for chemotherapy. Right. HIV infections and AIDS, because it's a retrovirus, and so is corona. SARS-2, interference with viral DNA synthesis from viral RNA using nucleoside reverse transcriptase inhibitors, nucleotide analogs. So we give an analog and then it makes erroneous information. Interferes with synthesis of DNA using nucleoside reverse transcriptase inhibitors. In AZT, that's how we use it. We use a thymine analog. All right, here's just another picture of just, like I said, we can't really kill the virus per se, like bacteria. We can just stop it from, we can kill it or we can stop it from reproducing or whatever. Viruses are a little bit trickier. We have to just figure out a way of making sure they can't get in. And once they get in, we can kind of mess with their ability to reproduce proteins. Now, interferon should be one of my last slides here, guys. I'm sorry, I've been going on for hours here. Human-based glycoprotein produced primarily by fibroblasts and leukocytes. All right, so remember, fibroblasts are those little cells that are living in your interstitial fluid. Interstitial fluid, you got the fibrin in there, and you've got some leukocytes hanging out, and that interstitial fluid, that matrix. And the leukocytes, leuko means white, cyte means adult cell. So these are produced by fibroblasts and leukocytes. And remember, fibroblasts can make cartilage, chondrocytes and chondroblasts, and they can revert back and forth, and they can make connective tissue and all kinds of cool stuff. But they can make these glycoproteins. So the therapeutic benefits of interferon reduces healing time and some complications of infections. And you can get drug therapy of interferon, or they can do research on how to stimulate your body to make more of this. Prevent and reduce the symptoms of cold and papillomavirus, bacteriomegalovirus, slows the progression of certain cancers, leukemias and lymphomas, and then treatment of herpes C, genital warts, and Kaposi sarcoma, which is an opportunistic infection, which is diagnostic for somebody with HIV and AIDS. All right. So if you have Kaposi's sarcoma and low T cell count, that's pretty much diagnostic for HIV and AIDS. So I'm going to go over the acquisition of drug resistance. Very important topic. Important for you to know it. And if you have a patient or family member or whatever who just doesn't want to take their antimicrobials, or they're like, oh, I feel better, I don't want to take these anymore, I want to save some for the next time I have it or whatever, you need to talk them out of that. All right, so adaptive response in which microorganisms begin to tolerate an amount of a drug that would ordinarily be inhibitory. Right? Due to the genetic versatility and variation, it's intrinsic and acquired. Right? So it can be Intrinsic through mutations that are turned on. It can be acquired through plasmid. Other bacteria will give it to them. And just remember, they become they're tolerated. So, I'm hoping none of you smoke, or if you are, you're going to stop. Or, if you're drinking, when you first start smoking, you're drinking, it might take a beer or two or a drink or two to get you to a happier place, let's put it that way. Or if you're first started smoking, you might, I don't even know, what, 5 cigarettes a day or whatever. And then you build up that tolerance to it. And with any kind of drug, medication, over and over again, your body adapts to it, and it should, right? It should be doing that. Same with the bacteria. So they either get inhibitory for it, or they'll find a way around it. It's adapt or perish. If something was coming at you in a threatening type of way, you would find a way to adapt and surpass it rather than succumb to it, correct? So acquired resistance, spontaneous mutations in critical chromosomal genes. So remember that your cells, bacteria cells, and specifically viruses, they all mutate. So these mutations can mean nothing. They can cause the organism to die. And there's viruses uploading the codes, telling you guys. So I have all these mutations, and they could be latent. They could be nothing for generations. And all of a sudden, something in the environment changes, and it turns on the epigenetics of this gene. And then this thing's able to survive. Everything around who doesn't have this epigenetic gene that wasn't even being used before dies, right? So that is survival of the fittest. That's how it works. You're going to have these mutations. They're not even expressed until something changes. All right. Acquisition of new genes or sets of genes via transfer from another species, so plasmids. All right, and these generally, I don't have a PhD in micro, but these generally have to be semi-related. All right, similar to species on Earth. We have to be semi-related, or we can't really, even though these have nothing to do with reproduction, they're not going to actually be able to transfer the information unless they're related. Because unless we're doing recombinant DNA, where we're like with genetically modified food or animals, we're going in and literally splicing the genes and inserting it scientifically, which I, have a problem with. If you're genetically modifying animals or food, I don't care what your rationale behind it is. I don't know if that's the best idea. Originates from resistance factors or plasmids encoded with drug-resistant transposons. So let's just say one bacterium has a resistance to erythromycin and the other one doesn't. But these two are going to dock to each other, and they have a sex plasmid to do something called conjugation. They actually insert the information. And remember too, they'll try to trick you. These plasmids have nothing to do with binary fission and replication. They're just going to go in and they're going to gene splice inside, fair and square, and then they can replicate. So plasmids have nothing to do with the ability to replicate as far as that DNA on the in the organism, right? And it's just showing you here. And read through this. So it's saying inactivation of a drug like penicillin by penicillinase. So these organisms can make enzymes or chemicals that inactivate whatever the process is that kills the organism. So they're going to adapt to it. An enzyme that cleaves a portion of the molecule and renders it inactive. It goes in and actually cleaves that side group. Remember we said before this was all the same? And we're going to change that side group there. All right. Number two, the receptor transports the drug is altered, so the drug cannot enter the cell. So we can go in, let's just see, we're talking about erythromycin or something like this, or cephalosporin, and it was something that they altered it so they could actually get into outer cell wall of gram-negative. Well, the gram-negative bacteria could say, you know what, I'm over it. And they're going to change their pore into the receptor site, so they can't even get in. Or the other one that's really kind of cool, specialized membrane proteins are activated and continuously pump the drug out of the cell. So we can get it in, but these cells, like making more mitochondria, they just make more pumps. They can pump it right back out. It's called an efflux pump. I put that there in red for you guys. And then we can change the drug binding site. So binding site on the target ribosome is altered, so the drug has no effect. So, we could change the binding site for tetracycline or erythromycin. It would no longer be effective, right? And then I said with the sulfa drugs, they're just going to go in, they're going to find another pathway. They'll find a way around it. All right. So natural selection and drug resistance. So I said before that these bacteria and even humans and everything on the earth, more or less, and specifically, like I said, viruses, we all have mutations. And I don't want to use the word silent mutations, but we have mutations, and you guys all, whether you believe it or not, or you think I'm crazy, you all have epigenetics, and you have, this junk DNA, they're calling it, that's there, and you just need to have it expressed. So that's changing your thoughts, environment, or whatever, without getting, like I said, into a lot of quantum physics. You have it there. So same with these bacteria and viruses. They have all this information there, and until it's needed or something on their environment changes, these are not expressed. So large population of microbes likely to include drug-resistant cells due to prior mutations in transfer plasmids. Now, they've never had to use these because they've never needed it, right? No growth advantage until exposed to drug. Or like, not to be crazy, but what if you had the ability to um, grow your hair at will, or if you were able to raise your temperature 5 degrees at will, no specific reason, and you never use that ability until all of a sudden it got super cold out and it was survive or perish. And you were able to do that. Everyone else around you couldn't. Or you're going to survive and they're not. So it's survival of the fittest. It's Darwinian. If exposed, sensitive cells are inhibited or destroyed, while resistant cells will survive and proliferate. So unexpressed gene, never had to do it. We change the environment. We expose it to a drug or a chemical or something. And then guess what? All these other cells or microbes die. You say, oh shit, I didn't even know I could resist those drugs or make EPHON pumps, or I could alter my receptor site. Or you know what? My receptor site was always a little bit different, but no one ever noticed, or it never caused a problem. But now that's kind of an advantageous thing, because now these medications can attach. And then realize that what's going to happen in survival of the fittest, all these non-drug resistance or ones that don't have the mutation, advantageous mutation, are going to die. So these are what's going to be producing. This one produced, it might reproduce with these, if these were actual cells or whatever, or if these were related, these cells, if these were E. coli or something, could give the plasmid information to this guy if they wanted to be nice about it. All right, so kind of read through strategies to limit drug resistance. Physicians have a responsibility for making an accurate diagnosis and prescribing the correct drug therapy. All right, so you want to be able to figure out, what's wrong with the patient, what is the likely cause, what is the best medication for it. And I'm hoping that, they're doing culture and sensitivity, or, not we're just throwing in these great big vials of stuff. They just put the causative agent in there. It has a reaction to something, antigen antibody reaction, or we could do a ELISA test or something like that, and it'll tell us immediately what to have, which is super cool. We don't have to wait for culture and sensitivity and gram staining and all that nonsense. Compliant with physician's guideline, the patient should take the correct dosage by the best route for the appropriate period. So dosage all the way through at the right time. That diminishes the selection for mutants and can resist low drug levels and ensures elimination of pathogens. So remember, it's going to kill out the weakest first, then the drug resistant ones, the weakest drug resistance, and what's going to be left are the strongest, most resilient, And that's when people stop taking the antimicrobials. And that's why we have part of the contributing to the problem of why we have all these stronger bacteria, because patients and we have to blame when we can partially blame, or we can partially discuss how physicians are indiscriminately giving antibiotics for viral infections, or how patients were non-compliant. There's a little bit of blame in all of that. Maybe someone took the medication and made them sick, so they stopped. There's there's always an exception. Combined therapy with two or more drugs together increases the chances of at least one of the drugs will be effective, and that the resistant strain of either drug um, will not be able to persist. So, like I said before, if you have something that's drug resistant, we're really not sure. So 1 and 1 is 3. So we'll give them more than 1. So we'll hit it different points. So long-term strategy. Proposals to reduce the abuse of antibiotic range from educating programs for health workers who require justification for prescribing certain types of antibiotics. All right. So we're not just indiscriminately just, oh, you have a sniffle, or you have some diarrhea here, just take this. Especially valuable antimicrobials may be restricted to their use for only 1 or 2 types of infections. So if we know you definitely have an infection of this or this, these are the 2 drugs. We're not going to mess around with something else. This is huge. The addition of antimicrobials to animal feed must be curtailed worldwide. So literally, the majority of antimicrobials in the world are going to they're going to livestock to stop infections so they grow faster, and and not to get crazy, but a lot of these feed homes, or these beef lots, or where these chickens are raised, are just completely beyond filthy and disgusting, and they just give these animals antibiotics because they're just living and breathing in bacteria and filth. Government programs that make effective therapy available to low-income populations should be increased. All right, because some people can't afford the medications, or they may have to share the medications with everyone in their family. They may only, I don't want to get into social economic stuff, but that may be all they have. Right? Vaccines should be used whenever possible to provide alternative protection. All right. Interaction between drug and host. So an estimated 5% of all persons taking antimicrobials will experience a serious adverse reaction to the drug or a side effect. All right, that's tetracycline there, by the way. See the brown? And that's possible. So it's very rare it happens on the first exposure to the drug, unless you've had exposure to something similar to that, so that you have antibodies to something that looks just like that. Generally, it's always the second time you take it, right? Majority said direct damage to tissue due to toxicity of the drug. Allergic reactions, disruption of the balance of normal flora, superinfections are possible. And that's what's going on with C. diff. Some of these superinfections, we're giving these broad spectrum antibiotics, or we're giving, a lot of these antibiotics for digestional things, and we're wiping out all the microbes. And, people have gastrointestinal issues, and IBS, and Crohn's disease. And the other thing, if you're watching this at all, you should be realizing that glycosamate and Roundup that's in the food, it's in the soil, it's in the root structure of these plants. You're consuming this, even if you're consuming vegetables, and it literally is destroying your microbiome, and it's actually destroying the desmosomes that hold all of your gastrointestinal cells together, so that things can't get into your digestive, great big particles can't get into the digestive tract. They should be absorbed through the microvilli and go through your hepatic portal vein into your digestive system. But they're literally breaking apart all those seals. And people say, "Oh, I have leaky gut syndrome." Well, yeah, no kidding. You're consuming pesticides, and it's even in the rainwater. It's so diabolical. It's in the the rain water. Thank you, Bill Gates. Whatever. Okay. And there's some super infections. All right. Potential pathogen resistant to drug, but held in check by other microbes. So this is drug resistant, but you've got enough microbiome in there that it has some competition, which is what you want. Normal flora is important in maintaining intestinal balance. You have to have your normal flora. And, please, when droning on about this, please start studying about your microbiome. It literally is way more important in your thoughts, feelings, moods, and activities than you can even imagine. All right. So drug destroys the beneficial flora. You wipe out all your microbiome. Now the pathogen, whatever's pathogenic, has no competition. Survival of the fittest. Now you have an overgrowth of whatever it is. And if this is Clostridium difficile or something like that, good luck getting rid of it, because now it is so prevalent. It's got no competition. It is so pervasive. It's going to be very, very difficult to get rid of. All right. There's some potential problems with some of these medications. All right. So considerations in selecting an antimicrobial drug, identify the microorganism causing the infection. All right. So we can use those ELISA tests or the antigen antibody reaction vials, put a little bit in, boom, we know what it is. Now, what's the most effective medication for that? So we know we're wiping out. Patients that take it the full course and now it's gone, we don't have to worry about it again. All right. Even if 10 of those bacteria still live and you excrete them out in your feces and someone else comes along and gets exposed to those, it could potentially propagate the whole thing all over again. Test the microorganism susceptibility to various drugs. And then here's the thing. It might work great in the lab on a petri dish or in that vial, but once you put it in the body, it changes things altogether. Right. And then the overall medical condition of the patient. Is the patient old, elderly? Do they have kidney or liver damage? So we can't give them something that may be more effective if it's going to tax the kidney or the liver, or is the patient pregnant? Is the patient already immune compromised? Do they have a bone marrow transplant? Are they aged? Do they have multiple myeloma? You have to take all these things into consideration. All right. So identification of the infectious agent should be attempted as soon as possible. All right, so you want to figure out what is it as soon as possible and wipe it out. You don't want to wait 3 or 4 days because that might have been the causative agent. Now you've got something else going on, maybe another bacteria, because you might be starting to get immune compromised or your body's busy. Now you have, I don't want to say a super infection, but you could have more than one microbe being the causative agent. All right, specimens should be taken before antimicrobials are initiated. So take the specimen before, give them the antimicrobials so at least you know what it was, and then you can do a baseline test to see did it diminish that specimen at all. There's no point in giving the antimicrobials and then taking the specimen because the antimicrobials may have affected what was originally the causative problem. All right. Here is the Kirkby-Bauer diffusion test, and I sort of did something like that in the video I made. So I just literally I did half E. coli and half Staph aureus, and then I just put different antimicrobials on there, same on each, and then I just measured the zone of inhibition. It didn't turn out really great this semester, but there was enough to give you guys the idea of what's going on. So what you really want to do is you want to figure out which is the biggest zone of inhibition. That would be the most effective. But you have to remember too, well, and just because it worked on the plate doesn't mean it's going to work in the body. Or you could say, well, that worked. Of course it worked, but it's very, very toxic. Is there anything else that works that'd be much more gentle? Or, this works and this works, they're both gentle. I'll give you both rather than give you this great big toxic one. Right. So there's reasons why you do that, just to give the infection. Now here's dog susceptibility, very, very, so this is the E-test diffusion. So this is, this strip is full of the antibiotic, put in there. And, towards the top, the concentration is greater, the zone of inhibition is still going to be there, right? Because the concentration is greater. But what you want to do is you want to go all the way down here and say, well, this totally worked, but the concentration of that medication at this level worked. So why don't we just pick something down here? It's less toxic. I mean, we could give them all that, but why? Let's give them this. It's less toxic on the body. Right. Let's get that. There's just some common, there's C. diff. Oh, no, that's Clostridium perfringens. That's for gas gangrene. Pseudomonas, that's the stuff that grows on your sponges. Salmonella, so chicken, eggs, related to E. coli. There's E. coli. There's a little Neisseria gonorrhea. Okay. All right, so just remember that just because it worked in the lab doesn't mean it's going to work in the body quite as well. All right. All right, that's it. I don't even know how long that went, but it felt like 2 hours, but we'll see here. 1 hour and 56 minutes. All right.